Okay, we are going to break apart the 4 categories of organic molecules. I'm going to talk about proteins first, and then I will get into the other 3 because proteins are probably the most extensive. So I'm going to break it apart. When we speak in terms of macromolecules in this course, we're talking about organic molecules. So everything is carbon-based. So proteins, carbohydrates, lipids, nucleic acids, they all contain carbon and are therefore considered organic molecules. So macromolecules, a generic term meaning large molecules. Sometimes they're referred to as polymers, so you may hear me say biological polymers. They are the same thing. Okay. So let's look at some generic background information or some vocabulary used referring to all 4 categories. Macromolecules and their component parts. When we talk about subunits, we're usually using the word monomer. And I will be using that word a lot, so that's something that you should get used to. A monomer is a subunit for a larger polymer. And anytime we join monomers, we do so by a dehydration synthesis reaction. So dehydration synthesis is how we join smaller subunits or monomers into polymers. And the picture does a good job of illustrating how that's happening. You've got your smaller monomers, 1, 2, 3. We're going to join them by removing water. And so you're seeing the H2O being taken out, hence dehydration, and we end up forming bonds between the monomers to build polymers. So anytime we're building, it's a dehydration synthesis. Anytime we're breaking, we're going to call that hydrolysis. So it is the opposite of the dehydration synthesis. We are going to be adding water in order to break larger polymers back into their respective monomers or their component monomers. So again, macromolecules, same thing as a polymer. They're broken down into smaller subunits using a hydrolysis reaction. Looking now specifically at proteins, the first of our 4 categories. The monomer or the building block of proteins would be the amino acid. So subunits equal monomers, same thing. Proteins are very versatile. They have several responsibilities within the cell. Some of those responsibilities include catalyzing reactions, and those proteins are specifically called enzymes. Proteins that are responsible for structure or composition are oftentimes just referred to as structural proteins. Some examples would be the flagellum of a bacterial cell. The flagellum is a structure used for movement, but it is composed of structural proteins called flagellin. We'll talk more about gene regulation when we get into the genetics component of the course, but when genes are switched on and off, those are controlled by, or that action is controlled by proteins. And then taking in nutrients. I know that in biology we studied transport proteins. They are embedded in the cell membrane and they help the cell get things in and out as needed. So all of these make up the role of proteins in the cell. So again, the monomer being amino acids. There are 20 different amino acids. And the general structure of an amino acid is shown. All amino acids have a carbon in the middle, a carboxyl group, an amino group, and a hydrogen atom. The carboxyl group being COOH, the amino group being the NH2. The R group, R meaning radical, sometimes referred to as the side chain, is the only thing that changes from one amino acid to the next. So of the 20 amino acids, they are exactly the same except for this group. So 20 different R groups exist. A protein's function depends on its shape, and that shape is dependent on the amino acid sequence. Amino acids are held together by peptide bonds. Okay, and a peptide bond is nothing more than a covalent bond between atoms in molecules. Remember that covalent bonds result from the sharing of electrons. But we're going to build a protein the same way we would build any organic polymer, by a dehydration synthesis reaction. So all we're doing here is showing specifically two amino acids. Here's the amino end, the NH2. This would be the carboxyl end of another amino acid. We take out water, an OH from one amino acid and an H from another. We remove those parts. We get water out, hence dehydration synthesis. The bond that results we call a peptide bond, and that's only in reference to building proteins, the word peptide. Okay, a bond is going to form regardless if it's a carbohydrate, if it's a lipid, but when we build a protein, we call that bond a peptide bond. So that word's unique to this category. Protein structure can be classified into 4 different levels, beginning with the simplest, primary structure. Primary structure is nothing more than the literal sequence of amino acids. In what order are the amino acids arranged? That's it. And that is dictated by the DNA. And so when we review transcription and translation, briefly, we looked at the messenger RNA and how it codes for which amino acids are brought in. Just that sequence is the primary structure of a protein. The secondary structure is going to look at how are the amino acids in that chain interacting. So how are maybe this amino acid and this amino acid interacting with each other? Because there will be weak attractions and even some repelling based on the polar and nonpolar nature of the individual monomers. So the secondary structure is the result of interactions among amino acids in the primary structure. And we could say it's a helical shape, or if it's a helical shape that results, we call those alpha helixes. Or a more pleated shape, we call that a beta pleated sheet, or just a beta sheet. And again, these structures, the helical shape or the pleated shape, are the result of weak hydrogen bonds between amino acids in their primary structure. So we get more and more complex as we move up. Tertiary structure is the fully developed three-dimensional shape. So you could have multiple helices, or a helix and a pleated sheet interacting, and that is making your tertiary structure. And so this is the fully developed three-dimensional shape. Typically, they're classified as either globular or fibrous. Globular proteins are usually your enzymes, your functional proteins. Fibrous are usually structural, so providing shape and support for the cell. By functional here, I refer to a working protein, or one that's ready to be used by the cell. When I say functional, referring to enzymes, those are ones that are doing or catalyzing reactions, either helping to build, helping to break. But a fully functional protein can be either globular or fibrous, and that does dictate the ultimate job of the protein. Is it speeding up reactions, or is it providing support within the cell? Quaternary structure, not all proteins achieve quaternary structure. Quaternary structure would be the interaction between two or more proteins that have reached tertiary structure. So you could have a globular and a fibrous protein interacting, and that would result in your quaternary structure. Okay, so I'll end this video and then we'll get into carbohydrates, lipids, and nucleic acids, because the last two are pretty small.